Biology Part 132, Mechanism of Contraction. When a muscle fiber contracts, each of the sarcomeres in its myofibrils shorten by a corresponding amount. This is achieved by molecular interaction between the two major components of sarcometer called thin filaments and thick filaments. The thin filaments consist mainly of protein called actin and are anchored at one end to the Z disc. They are only filaments in I band regions of the fibril and overlap with the thick filament which occupy the A band. The thick filaments consist of a second protein and called myosin. Within the H zone, only thick filaments are present. Measurement taken from electromicrograph of contracted muscles show that the width of the H zone decreases while the lengths of the filaments remain unaltered. It follows that during contraction, the filaments slide past one another, as shown in the figure. And this proposed mechanism gives rise to the sliding filament theory of the muscle contraction. The forces which produce contraction depend on the formation of cross bridges between the actin and myosin molecules. Actin molecules are globular proteins which associate to form a twin-stranded helical structure, which is basis of the component of the thin filaments as shown in the figure. Each actin molecule has a specific binding site for myosin. Myosin molecules which are large and have a pair of globular heads attached to a long fibrous tail containing flexible hinge. The thick filaments are produced when the trails of many myosin molecules associate together, leaving their globular heads projecting outwards. Cross bridges between the filaments are formed only when ATP is available. ATP combines the individual myosin heads and is then dehydrolyzed, the production of reaction being temporarily retained by myosin head. This hydrolysis caused a change in the shape of head so that it is now able to bind the actin filament. As soon as this happens, there is a second change in shape, which results in the release of ADP and inorganic phosphate. This change represents the power stroke of the system, and it generates mechanical energy which is used to pull filaments along. During contraction of a muscle fiber, this cycle of events is repeated many times as the heads of myosin molecules from the thick filaments walk along the actin molecules of the thin filaments. Initiation of muscle contraction. ATP is not only the requirement of contraction in living muscle cells. Ca2 plus ions are also needed. For their absence, contraction is blocked. This is explained in the presence of additional proteins in the thin filaments, namely troponin, and we can see them in the figure as well, as explained there. The tropomyosin, in fact, is during an inactive period. The molecules of these proteins lie along the actin filament in such a way that all of the myosin binding sites are covered, making the formation of cross bridges impossible. Ca2 plus ions bind the necessary accessory proteins and twist them so as to expose the binding sites. This remarkable mechanism provides the essential link between the motor nerve pulses and muscle contraction. Each skeletal muscle fiber is an electrically excitable cell receiving impulses across the synapses called neuromuscular conjunctions. At these junctions, the transmitter substance acetylcholine causes depolarization of muscle cells membrane of the sarcolemma and may trigger in all or one action potential.